The Wolbachia program is specifically focused against the dengue virus, but is likely to work against Zika virus as well. Wolbachia is a bacterium that occurs in many species of insect naturally, but it doesn't occur in the vector of dengue fever, which is Aedes aegypti. So to make the Wolbachia program work, we need to put the bacterium inside the mosquito, and when the bacterium's in there, it prevents the mosquito from transmitting dengue virus. So what we do is breed a lot of these mosquitoes in the laboratory that all contain Wolbachia, and then release them in the wild. And they have a mechanism which allows the spread of Wolbachia through the mosquito population, which is caused by the presence of Wolbachia. And that's called cytoplasmic incompatibility. And it gives the Wolbachia female mosquitoes a reproductive advantage because they can mate with an infected Wolbachia mosquito or an uninfected mosquito with no Wolbachia, and all of their offspring will be viable and contain Wolbachia. But a mosquito female that doesn't have Wolbachia, if she mates with a Wolbachia infected female, then all of her progeny will die. So it's better to be infected, and then you can mate with either type of male. And this mechanism allows the bacterial infection to spread throughout the mosquito population. And eventually all of the mosquitoes within the release area should contain the Wolbachia bacterium. Our laboratory is interested in finding the best Wolbachia strains for release in the field, ones that will be able to survive out there. Often with lab-reared insects, they might have some defect that doesn't allow them to compete with the mosquitoes under natural conditions. So we do lots of experiments to make sure that our Wolbachia mosquitoes are fit. And there are many different strains of Wolbachia, and some of them affect the mosquito to different extents. Some of them make the mosquito very sick, or not lay as many eggs, or not live as long. And they may not be very good candidates for release in the field. It means that there is hope for a long-term control option that can be put into place in an area and then and left there to do its job. Some other techniques are shorter term, and insecticide use, as we've seen, is coming to an end in some places in its usefulness. So hopefully with the Wolbachia approach we can cover many areas, and then as they become established move on to new areas. And because the Wolbachia drives itself through the mosquito population, we don't have to keep releasing all of the time in the same area. The Oxitec method of mosquito control for dengue fever involves a genetic modification of the mosquito so that they're carrying a gene that causes their offspring to die, except if the offspring are reared in the presence of an antibiotic. So the scientists can rear these mosquitoes in great numbers in the laboratory in the presence of an antibiotic. Then they release males into the field. The males mate with the wild female mosquitoes, but all of the progeny don't live to become adults, and that causes a suppression effect of the mosquitoes, so a great reduction in numbers. It has great potential in some areas, probably on islands or remote locations where other mosquitoes are not re-invading, because this is a technique that doesn't drive itself through the population. For it to be successful, the mosquitoes carrying the gene have to continuously be released while there are wild mosquitoes in the area.